Hello, my name is Alec. I'm a master's student in Dr. Ghosh's lab. My project is exploring a role for the Hippo pathway in a non-canonical apoptotic program. Our lab works on a non-canonical apoptotic program, the nematode C. elegans, that we call CCE, or compartmentalized cellular elimination. A little bit of background on program cell death. Program cell death is an important mental developmental event. In humans, we start with webbed fingers, but the cells between our fingers die in a programmed way. Neurite pruning is also an example of programmed cell death. There are multiple types of programmed cell death, with the most characterized being apoptosis, which is characterized by cellular shrinking, rounding, and chromatin condensation. Genetically, apoptosis requires caspase protease, with the main regulator being human caspase 3. There's another type of programmed cell death, which is linker cell death, which was first described in nematode C. elegans. Elimination of morphologically complex cells is poorly understood. The human body is made up of many types of cells, many of which have different protrusions and extensions that go into different microenvironments. And within these cells, there are different subcellular architectures that influence how they die. The Hippo signaling cascade is an evolutionary conserved signaling network that regulates many things. For example, the Hippo signaling pathway regulates cell proliferation, apoptosis, movement, and cell fate. This regulation of this pathway is implicated in many diseases such as cancer. Recent work has implicated it in neurodegenerative diseases. We study this system in the nematode C. elegans because it has an invariant lineage, it's transparent, genetically tractable, has a short life cycle, and are self-fertilizing. In C. elegans, the genetic program for apoptotic cell death was first described. Here I'm showing you the classic stages of compartmentalized cellular elimination, or CCE. As the tail spike cell begins to die, the soma or cell body rounds up like a standard apoptotic cell. The proximal process undergoes beating, while the distal process retracts into itself. At the end of CCE, two fragments remain, which is the soma and the distal process, both of which are consumed by different neighboring phagocytes. So my central question is the Hippo pathway involved in our novel program of cell death, CCE? I look at this question through two main ways: looking at the embryo where CCE takes place, and looking at L1s where the tail spike cell should have already died and been cleared. In AGL44 TEAD mutants, the soma persists after the threefold embryo stage and into the L1 stage, where the tail spike cell should already be dead. After I looked at AGL44, I looked at YAP1, and the YAP1 mutants showed a mainly process elimination defect. Interestingly, the tail spike cell does not show YAP1 expression. However, the neighboring phagocyte of the process does show nuclear YAP1 expression. Some of our future outstanding questions are: does AGL44 function cell autonomously? To look at this, we will perform cell-specific rescue and look at the expression. Other outstanding questions that we have are: what is the role of YAP1s? What is the role of YAP1 in CCE? We will look at cell-specific rescues to determine whether YAP1 functions cell autonomously, as well as trying to figure out whether these fragments that we see of the tail spike cell are internalized by the phagocyte. We will look at this with a phagocytic marker.